There are a couple of special codons that we always need to remember. The first is the start codon. If you look in the genetic code table, you'll find the start codon right here. The start codon across all living cells is AUG. The ribosome will recognize this codon, AUG, as the place to begin translation. It will actually place a methionine at the start codon. And it will then continue to translocate down the length of the mRNA and continue translating and producing a polypeptide until it hits a stop codon. There are 3 different stop codons in the genetic code. The first one is UAA, the second one is UAG, and the third one is UGA. If the ribosome encounters any of these 3 codons, it will stop translation. So, we know from the genetic code that the third position of a codon is somewhat resilient to mutation because a lot of amino acids can have any base in that position and will still have that particular amino acid placed at that particular location in the polypeptide. The bases in the first and second positions in the codon are quite different. When mutations occur in DNA, and those mutations alter the bases found in the first or second positions in codons, they will change the particular amino acid that gets placed in that location. Remember, if you change the order of the amino acids in a polypeptide, you will change that protein. Even a single amino acid difference will have enough effect on how that protein folds as to change its function. If you change the amino acid sequence, you will change the protein and you will change the phenotype, the collection of traits that that organism has. We spoke about Tay Sachs disease earlier in the semester when we were talking about organelles. We said that Tay Sachs disease, which is a deadly disease in young children, Tay Sachs disease is a lysosomal storage disease. And Tay Sachs is caused by a single amino acid change in a particular gene. Sickle cell disease is another good example of a really terrible human disease that's caused by a change in a single amino acid. There's another disorder in humans called FOP. That stands for fibrodysplasia ossificans progressiva. This is a particularly rare disorder in humans, but it causes an absolutely devastating disease. This young lady here in the photo is an FOP patient. I want you to notice her posture. In FOP, the effect of that single amino acid change in a particular gene causes a disorder where even the slightest bump, the slightest injury to the muscles and connective tissue around the skeleton will trigger that tissue to become inflamed and to ossify. The muscle and the connective tissue will turn to solid bone with any kind of slight injury. The way this woman is standing is dictated by the fact that she has now grown bone in places where it does not belong. She can no longer bend her legs in any other position besides the one she's in. She can no longer raise her hand and bend her wrist because it's now locked in that position with abnormal bone growth. Over here, you're looking at a skeleton that was donated by a person who suffered with FOP. I want you to look at all the extra bone on this skeleton. In the past, people who have inherited this genetic defect and who have lived with FOP, they have rarely lived beyond 30 years old. What eventually tends to happen is new bone formation will impact their rib cage, and it will cause them difficulty in breathing, and they essentially suffocate. The good news for FOP sufferers is that about 10 years ago, the abnormal gene was discovered, and new treatments have actually been introduced, although they're still in experimental trials right now. So, it's possible for the order of the bases in the DNA molecule to be altered. It can be altered by chemicals, it can be altered by radiation, it can be altered by certain types of infection and chronic inflammation. It can also be altered just by mistakes that are made by the enzyme that performs DNA replication. When mistakes are made and a change occurs in the order of bases in the DNA molecule, the cell has an opportunity to find that mistake and repair it. But sometimes these mistakes persist and they become permanent. That is a mutation, a permanent change in the order of bases in the DNA molecule. What we're going to do for the rest of the lecture today is talk about two very different types of mutations. One is called point mutation, and the other is called chromosome level mutation. We're going to talk about 4 different types of point mutations: silent, missense, nonsense, and frameshift. And we're going to talk about 4 different kinds of chromosome level mutations: inversion, duplication, deletion, and translocation. When we use the term point mutation, we're saying that there has been a change in a single base in the DNA molecule. Now, sometimes it's a substitution type. In other words, one base has been substituted for another base. Sometimes a single base will be added in to the DNA molecule, a base that shouldn't be there. That's what we call an addition. And sometimes a base will be completely eliminated from the DNA molecule. That's what we call a deletion. If you look at the diagram at the bottom of the slide, over on the left, we're looking at a DNA molecule, a very short DNA molecule, to be sure. It's double-stranded. You can see the bases are paired in a complementary manner. Notice the 2 strands are anti-parallel to each other. 5' and 3' ends are opposite on the 2 strands. During DNA replication, remember, each of these strands will be used as a template to build new DNA. They're showing us that this top strand is right here now, and new DNA is being built in red. The DNA polymerase enzyme matched a T with this existing A T with this A, a G with C, so far so good, an A with T, and then it made a mistake. It placed a T where there's a G in the original DNA. And then it went on to do correct pairings, C with G, G with C. The cell has an opportunity to catch this mistake. We don't talk about proofreading or repair mechanisms in this course. You will talk about those in genetics. But for now, understand that these two bases will not pair correctly. Remember, A and T and G and C will hydrogen bond with each other because they are correctly paired. But T and G will not correctly pair, and therefore they will not hydrogen bond with each other in the normal way. And that's going to create a little bit of a distortion in the DNA molecule. The cell can catch this in proofreading, and it can repair it. But sometimes it will miss it, and this problem will persist. Now, remember what's going to happen. DNA replication occurs when cells are preparing to divide. The original parent cell had this DNA. One of the daughters is going to inherit this DNA. If this mistake is not fixed, that cell has inherited an abnormal piece of DNA, and when it goes to divide, it's going to replicate this DNA. And anytime DNA is replicated, the two strands will be separated and used to build new DNA. So this gray strand is shown here, and this red strand is now shown here. So this daughter that inherited this abnormal DNA is now itself dividing, and so it's replicating its own DNA. This piece, the old DNA and the new DNA, are perfectly fine. In fact, you can see they are an exact copy of the original DNA. But down here, we had a strand that had a change in it. The enzyme is going to come along, and it's simply going to pair up what belongs with that T. It's going to put an A there. What's happened at that point is the change in the order of the bases has become permanent. You now have a mutation. No longer do we have two bases that don't belong together. Now we have two bases that do belong together, right? A and T. They're gonna hydrogen bond perfectly normally with each other. Sorry, I just realized I'm out of your frame of view. This A and T, they belong together. The T shouldn't be there. The T was put there by accident when the original parent cell divided. But now this daughter, she's got the T in her DNA. Her cell will assume the T belongs there, and it will match it with an A. Now this is a correct base pairing, A and T. It doesn't belong there, but it's a correct base pairing. It's stable, and it will persist. It will not be caught during proofreading anymore. There's no chance that this is going to be repaired now. This is mutated DNA. The first kind of point mutation in our list is the silent mutation. In a silent mutation, we have a base substitution, again, one base for another, but the change in the base does not change the order of the amino acids in the protein. Remember, this is that third base location in codons oftentimes. Those are silent. There's a change in the order of the bases, but that change does not cause a change in the amino acid sequence. The change in the order of the bases changes the genotype, but it does not change the protein. So it's a silent mutation. It does not cause a change in the phenotype of the organism. Missense mutations are different. They are also substitution type point mutations. One base was substituted for another. But this time, the change in the bases leads to a change in the order of the amino acids. Missense mutations often occur at the first and second base in the codon, or in the third position for a couple of the amino acids. Again, we've got a change in the order of the bases in the DNA, so that's a change in genotype. But this time it also changes the protein. We've changed the order of the amino acids, so we have changed the protein and changed the phenotype. Nonsense mutations are a very particular type of substitution. Again, one base for another. What happens in a nonsense mutation is the change in that single base produces a stop codon that didn't used to be there. In other words, what we call an early stop codon. Before the mutation occurred, there was a codon that coded for an amino acid. After the mutation occurred, that codon became a stop codon. Remember, anytime a ribosome encounters a stop codon, it's going to stop translating the RNA. That means, depending on where this nonsense mutation occurs, that means the polypeptide, that string of amino acids, is going to be shorter than it should be. So a nonsense mutation changes the genotype, and it causes a massive change, typically, in the protein. Many amino acids will be simply missing because translation stopped too early. The last kind of point mutation is called a frame shift mutation. This type of mutation is not a simple substitution. Now we are either adding in an extra base or we are deleting a base entirely from the DNA. And when that happens, it's going to shift what's called the reading frame of the codons in the RNA. If you add in an extra base into the DNA, or you take a base out, delete it, now the groups of 3, the codons in the RNA, are going to shift by 1 base. Again, that's going to change the genotype, but it's going to also cause a massive change in the protein. If you shift the reading frame in the RNA, you have changed every single codon from that point onwards. And that's going to cause a massive change in the protein, a massive change in the phenotype. Many human diseases are caused by point mutations. We talked about a couple of diseases that are caused by single amino acid changes. And many of those single amino acid changes are caused by point mutations, one base among 3 billion base pairs. We're going to switch gears here, though, and we're going to talk about a completely different type of mutation called a chromosomal mutation, or a chromosome level mutation. We are no longer talking about single base point mutations here. In a chromosome mutation, or a chromosome level mutation, entire portions of a chromosome are going to change. And there are different methods for how those change, and we'll talk about each one. Chromosome level mutations are massive mutations because we're not talking about a single base here. We're talking about whole segments of the DNA molecule, whole segments of the chromosome. The first kind of chromosome level mutation is called a chromosome inversion. In this type of mutation, a piece of the chromosome is going to break away. It's going to break off. It's going to flip 180 degrees, and it's going to reattach to the same chromosome. Look at the picture down on the bottom. They're showing us a particular chromosome labeled chromosome A, and it's got regions on it, A, B, C, D, E, F, G, H, I. Maybe these are individual genes on this chromosome. A segment of the chromosome in the middle, in the internal regions, has broken away. It's going to flip over 180 degrees and reattach. Now you may say to yourself, well, it's not that the DNA has changed in terms of the individual bases. All of the individual bases are still there, but the sequence has changed because the sequence of bases should have gone through gene A, through gene B, through gene C, and into gene D, E, and F. Now it goes from C to F, E, and D. Remember what we said about genes. They contain code to build a protein, and they contain regulatory code. What we've done here is we've literally reversed certain gene sequences. Genes are not intended to be read in any direction. They should be read in one particular direction, and we've now essentially reversed them. You can imagine, not only are multiple genes involved in these, but they've essentially been rendered badly damaged, if not completely non-functional. Chromosome inversions, like all chromosome-level mutations, cause very significant changes to the organism. The next kind of chromosome-level mutation is called a chromosome duplication or a chromosome deletion. These are two different kinds, but they're often grouped together because they're similar. In chromosome duplication, a segment of the chromosome is actually going to be duplicated. In other words, entire regions of DNA are going to be replicated abnormally to produce a doubling of that region of DNA. Related to this is what's called chromosome deletion. And this time, entire regions of DNA will simply be deleted from the chromosome, removed and not replaced the way they were in the inversion. When a duplication or a deletion occurs, again, you've got a massive change, a massive effect to the organism because multiple genes can be involved. Sometimes genes are completely eliminated, which would mean no protein was produced when it should be produced. And sometimes you've got a duplication of genes, so you're going to get much too much protein made from that gene than the organism would normally produce. Both of these are detrimental to the organism. You're going to hear me say proteins are powerful things. Not having protein that you need as a cell is very damaging. But so is having too much of that protein, even if it's a perfectly normal protein in its structure. If the cell ends up producing more than it should, it can have adverse effects. Next on the list is chromosome translocation. In this type of chromosome level mutation, a piece of a chromosome is going to break off and then reattach, but this time to the wrong chromosome. Remember, in an inversion, the piece that breaks off flips over and then reattaches. In a translocation, the piece is going to go away from its original chromosome and reattach to a different chromosome. If you look at the drawing, you can see a green chromosome and a pink chromosome. And there are boxes drawn around the regions of these chromosomes that are going to translocate. This piece of the green one breaks off, this piece of the pink one breaks off, and they essentially change places. There are certain chromosomes in humans that are particularly susceptible to this happening, and we don't exactly know why. But as you can see from the drawing, what this is going to do is create two brand new chromosomes. It has reordered the genes, and it has absolutely altered two chromosomes. That might lead to gene sequences that are transcribed too often or transcribed not often enough, because these particular genes are now in a brand new regulatory environment. Massive, massive changes for the organism. The last thing I want to say today to finish up our discussion of mutations is this. Sometimes it's hard for us to understand in our minds what the effects of mutations are. We generally understand that mutations are sometimes not a problem, right? Silent. And sometimes they are a problem. Missense, nonsense, frame shift, chromosome level. But the other thing we have to be able to understand is that there's a difference between a mutation affecting the organism in which it occurred and the mutation affecting future generations. The consequences of mutations are dependent upon the types of cells in which the mutation occurs. Remember, when we looked at the karyotype, we said there are chromosomes that contain genes that help build the ordinary body cells. And then there are chromosomes that help build the cells that allow an organism to reproduce. Reproductive stem cells are the cells that create the gametes, the eggs, and the sperm that will go on to create brand new organisms. When mutations occur, if they occur in the somatic cells, skin cells, liver cells, pancreas cells, blood vessel cells, stomach cells, brain cells, if they occur in the somatic cells, those types of mutations can be passed on, again during cell division, to subsequent generations of cells in that organism. But they will stay in that organism. When that organism dies, the mutations will die with it. The consequences, therefore, will be to that organism. And they'll vary depending upon the type of mutation. If, however, you mutate reproductive stem cells, now you've altered the DNA in the gametes. That DNA and those mutations are going to be passed on during cell division to the next generation of sperm and egg, but they will also be passed on to the offspring, because it's the gametes, the egg and the sperm, that literally create the next generation. So the consequences of a mutation in the cells that make gametes include the possibility of passing on new traits, new phenotype, to generation upon generation of offspring. Somatic cell mutations live and die with that one organism. Reproductive stem cell mutations get passed on